Hi everyone, my topic is going to be on how leucine-rich protein intakes affects skeletal muscle protein synthesis through the mTOR pathway. I am choosing to work on mTOR pathway because it has received a lot of attention. If you are interested in longevity, you would hear about mTOR quite a bit. I am working on only the protein synthesis piece of it because I'm doing a graduate program in nutritional science. So I'm interested to learn more about how dietary protein intakes affects how we build muscle. And we actually have learned a little bit about mTOR in our chapter 7. We didn't touch a lot on it, but so this presentation is a more granular view of how protein intakes affects how we build muscle through the mTOR pathway. So how is mTOR relevant? So the mTOR pathway is a signaling growth pathway. It exists in different tissues, and in different areas it actually has different activities. In my presentation, we're only going to touch on the anabolic pathway regulating skeletal muscle protein synthesis. And who does it affect? It actually affects many people, especially people who are concerned about bodybuilding or who wants to build muscle. And also it affects our elderly populations because sarcopenia is actually pretty common in the elderly populations. And how much muscle our elderly person has is actually a predictor of hospitalizations and all causes mortality. So here are some historical perspective on mTOR. So it started out in 1964 when two Canadian scientists, George Nogrady and Dr. Stanley Scorina, they discovered an antimicrobial agent called rapamycin, as they were noticing that people were getting tetanus, and they handed the information and discovery over to Sharon Siegel. And being unaware of rapamycin's potential, Siegel noticed that rapamycin was exhibiting really amazing antifungal property, especially inhibiting the growth of yeast and mold. And because of that, another biochemist, Michael Hall, he discovered the protein target of rapamycin in yeast cell. And later on, another scientist, Dr. David Sabatini, he discovered the mechanistic target of rapamycin, and they call it mTOR because of the homology to the yeast TOR genes. And by 1999, the FDA has approved rapamycin as an immunosuppressant. And since the discovery of mTOR, it opened many doors for many cancers and anti-aging researchers. There are many researchers and reviews articles out there about mTOR because researches on mTOR are still relatively new. I have mainly chosen the ones that are more recent because the older reviews articles, there were still a lot of things that they didn't fully know how it works. So researchers and reviews article that after about year 2020 gives me more information. So that's how I chose my reviews. And about the mechanism. So mTOR is a series threonine protein kinase. It has two complexes. So it is mTOR1 and mTOR2. In this presentation, we're going to focus on mTOR1. So it is a growth regulator, and it links between the availability of nutrient in the environment and the control of some of the anabolic and catabolic processes together. And mTOR1 responds to amino acid, oxygen availability, energy level, and growth factor. So mTOR1 regulates cell growth and metabolism through the modulations of protein and lipid synthesis, lysosome biogenesis, and autophagy. In Dr. David Sabatini's words, it is built to detect if there's any presence of a poor condition. So there are many possibilities of good conditions, such as having enough nutrients like glucose and oxygen and amino acid and all that things. But when there's a poor condition, such as amino acid, then mTOR kind of shut things down to protect against the poor condition, because it makes sense that if we don't have enough amino acid, we really shouldn't be making protein synthesis. And so this regulations of mTOR activity is associated with diseases including diabetes, cancer, and neurodegenerative disorders. So this is how mTOR1 looks on animations. The complex is made of many different proteins that have both activating and inhibiting functions. There are two sensors to sense leucine. Leucine is an essential amino acid that our body cannot make, so we will only have enough leucine if we eat enough proteins. And one of the leucine sensor is called LAS. It is a GTPase accelerating protein, GAP. So it has four different parts. It has rad A and rad B. They are GTP loaded, and while rad C and D is GTP loaded. So when we sense leucine, the rad A and rad B will interact with rad C and D to form a heterodimer, and the leucine will induce the exchanges of GDP by GTP in rad A and B, and because of that, it converts the GTP to GDP in rad C or D, and that results in the rad heterodimer that eventually activates mTOR1 on lysosome. So there's another leucine sensor, which is called SIRTRIN2, which stands for stress response protein 2. It inhibits GATA2, and GATA2 inhibits mTOR1 activity through inhibiting GATA1. So leucine inhibits the interaction between SIRTRIN2 and GATA2, and that leads to mTOR1 activation. So in the previous slides, you may be thinking that, okay, when we are activating mTOR, it says activating lysosome. So how does that leads to protein synthesis? So it turns out that lysosome is more than just a recycle bin. It is found to be an intersections of cellular growth and destructions. So high mTOR1 promotes amino acid exports from lysosome when amino acids are available, and low mTOR activity inhibits amino acid from leaving lysosome. So that's how it plays a role in protein synthesis. So our mechanism tells us that even if we have all other nutrients, if we are missing amino acid, specifically leucine, mTOR1 will put a pause on protein synthesis. So my primary article, they conduct a research study with a group of 50 age 50 to 70 healthy adults. So they have the intervention group to drink a protein powder drink with 20 grams of proteins with 2,000 milligrams leucine, and do 60 minutes of resistance training for 12 weeks, with the placebo group doing the same amount of exercise, but drinking a placebo drink, which is just a sugary drink, and they want to see if there's any difference in the result. So 60 minutes of resistance training sounds intimidating, but they did talk about what they do in the training program, because these people, older people, so we can see that the resistance training is really not that intimidating. And out of the 60 minutes, it includes 10 minutes warms up and 10 minutes cools down. And some of the things they do is really just to sit and lift their legs, do sit and stand, do a body twist, do a push-up against the wall. So really nothing too intimidating. So after the 12-week study, they compared the result between the two groups, and they found that the increase in lean body mass and skeletal muscle mass was significantly larger in the intervention group who drinks the protein shake. And they also generally do better in terms of muscle strength performance. And what are muscle strength performance? They measure that by seeing how many reps of push-up they can do, or they want to see their hand grip strength, and they did significantly better in plank performance, while other functional tests are roughly the same. And the other functional tests include things like how many reps they can do sitting in a chair and standing up within 30 seconds, how many bicep curls they can do. So the groups that were drinking and having enough protein and leucine, they were gaining more muscle mass. But my other takeaway from this study is that no matter which group the people were from, after the 12 weeks of the resistance training, overall the strength performance were much better than the baseline, and even if they weren't drinking protein drinks. So that kind of tells us the importance of being active and just doing things and using our muscle to keep the muscle mass. So here are my questions for us to think about. Thank you for watching.